My name is Richard Logan, and the chemotherapy drug that I'll be talking to you today about is called Taxol. Taxol belongs to a class of chemotherapy drugs called plant alkaloids. It was isolated from the bark of the Pacific yew, and it proved to be very effective at inhibiting cell division in cancerous cells. As you all know, the cell cycle consists of 4 parts: G1, S, G2, and mitosis. And as in mitosis, it involves interphase, metaphase, anaphase, telophase, and cytokinesis. So Taxol's function is Taxol blocks the cell cycle in its G1 or M phase by stabilizing the microtubule cytoskeleton against depolymerization. Taxol works by stabilizing the microtubule polymer, as you'll see to the right. The growing ends of microtubules are protected from this depolymerization by a cap of GTP tubulin dimers. Taxol binds directly to the B tubulin subunits within heterodimers. As you see right there where the green arrow is pointing to. GDP tubulin dimers have a slightly different conformation that makes microtubules contain them less stable, favoring depolymerization. But with Taxol associating along the entire length of a microtubule, Taxol can help prevent microtubule depolymerization even if the GTP cap disappears by preventing compaction of GDP tubulin dimers, which normally causes microtubule catastrophe, which you see right here to the right, bottom right. Like normal cells, cancer cells also undergo interphase and the 4 stages of mitosis. The only difference is that in cancer cells, the mechanisms that regulate systems, such as the one that makes cells proceed through the cell cycle, are defective, which means that these systems are activated regardless of these control mechanisms. And the systems that normally stop cells from proceeding are inactivated, meaning that cancer cells don't divide faster, but they do not stop or take breaks between life cycles like normal cells do. They keep dividing. As for Taxol, when Taxol is applied to the cells, the spindle fibers are interrupted and the cell remains in metaphase. After the cell is in metaphase for a while, to put it simply, it interferes with cell division and ultimately leads to cell death in those fast dividing cancer cells. As in normal cells, cell division is under control by the checkpoints and everything occurs only when required. Taxol's major side effects. Taxol is primarily used for the treatment of breast, ovarian, and lung cancer, as it is proven to be the most successful in treating these type of cancers. But the limitations of Taxol does not know the difference between cancer cells and normal cells, although it is likely to damage cells that are at rest, which are mainly normal cells. It affects healthy body tissues where the cells are constantly growing and dividing, such as hair, which is always growing, bone marrow, which is constantly producing blood cells, the skin and the lining of the digestive system, which are continuously renewing themselves. And all this results in low blood counts, mouth sores, nausea, diarrhea, and hair loss. Most side effects disappear once the chemotherapy attacks are over. Normal cells will be able to replace or repair the healthy cells that were damaged by Taxol. So my question for y'all today is, Taxol targets tubulin in the cytoskeleton and inhibits mitotic spindle assembly. But the goal is to inhibit cell division in cancer cells, which often reproduces rapidly. Considering this mechanism of action, would Taxol be an effective drug to treat a bacterial infection, since bacteria also reproduces rapidly?